Hello everyone, I am Professor S.R. Kale from KKO College of Food Technology, and I am here with the shortest topic and the simplest topic of biotechnology, that is gene cloning, which is considered as a base for the whole biotechnology cloning concept. We will begin with the introduction. We all are familiar with the word gene and the cloning, but here we can study the gene cloning. See, gene is a unit of inheritance. Means whatever the hereditary characters that are completely forwarded by the genes, and a segment of DNA that provide instruction for synthesis of a specific protein or a particular type of RNA. Means gene is a segment of DNA. What is gene? It is exactly a part of DNA, or we can call it as a segment of DNA. It is providing instruction for the synthesis of protein or a type of RNA into the body. Definitions: gene, a segment of DNA which is responsible for inheritance and expression of particular character. What is gene? It is a segment of DNA and which is responsible for the forwarder of inheritance character and expression of particular character into the body. The definition of clone: a clone refers to a group of organism, cell, molecule of objects arising from single individual. Simply, clone is what? It is referred to a particular group of anything, like any organism, cell or molecule of any object which is arising from a single individual. Now we will see gene cloning process in detail. Gene cloning can be explained with the help of following given points. First one is generation of DNA fragment. Now we will see what happened in case of generation of DNA fragment. It can be done by using restriction endonuclease digestion, cDNA synthesis, PCR and chemical synthesis. Next is insertion into the clone vector. It can be done by using ligation or blunt end or cohesive end. See what happened in our first case, that is generation of DNA fragment. This generation of DNA fragment is done by using restriction endonuclease digestion. Means here we are breaking that fragment from to the host, or we are using our cDNA technique, PCR technique or any chemical synthesis technique for the separation of DNA fragment. Then we are doing insertion into the clone vector. Means whatever the fragment we are separating, that we are going to insert into our clone vector, and this can be done using ligation of blunt end or cohesive ends. Next, third step, that is introduction into host cell. It can be done by using different three techniques, that is transformation, transduction and transfection method. That is insertion of a vector cell into the target cell is usually called as transformation. For bacterial, transfection process. For eukaryotic cell, although insertion of a viral vector is often called transduction. Our first step is selection or screening. It can be done by using hybridization or PCR method. So guys, here we are completed with our gene cloning topic. Thank you everyone.